just before we studied about glycolysis, gluconeolysis, like that. So in fasting, around 6 hours, we know that our stored glycogen is taken for energy, and around 27 minutes of intense workout, this will go, and the fat starts to be break down for energy. So that's what we are going to study now, lipolysis. So what happens is that, assume that this is our fat cell. In that, the fat is stored as triglycerol. Okay. Now the process is the breakdown of triglycerol to fatty acid, separate it to fatty acid and glycerol. Okay. So what exactly happens is, when we are in fasting stage, our glucagon increases and insulin decreases. Okay. So glucagon is the substance which breaks down fat and glycogen. Okay. So first the pancreas will release glucagon, and it is coming to the fat cells and caught by the 7-path receptors. Simultaneously, the adrenal gland will produce the epinephrine, and it also gets attached to the fat cell through this 7-path receptors. So what is the use of these two things is that it activates the G stimulatory protein inside the cell. It's written here intracellularly. Okay, that means inside the cell. And this G stimulatory protein's role is to stimulate, as the name suggests, is to stimulate what? Adenylyl cyclase. Okay, so this G-stimulatory protein stimulates this, and that cycle, there is a cycle which converts ATP into AMP. ATP is adenosine triphosphate and AMP is adenosine monophosphate. Okay, so what happens next is that this AMP, adenosine monophosphate, activates. There is a battery-like thing, that is protein kinase A. It will activate protein kinase A, and then it has got this AMP inside, and the phosphate part is getting to hormone sensation lipase. Okay, so before in glycolysis also we studied adding the phosphate is known as phosphorylation. Okay, in glycolysis also the same. So it adds the phosphate. And what happens next is that when this hormone sensational lipase is getting this phosphate, it works like a cutter, splitter. It splits the fatty acid and the glycerol bond from triglyceride, and this glycerol bond and fatty acid moves in the circulation, and this glycerol bond will go to, we previously studied the process of glycolysis, how glucose becoming pyruvate and how the fat going to glycerol aldehyde 3-phosphate. Okay, there is an enzyme here which converts this coming glycerol to glycerol aldehyde 3-phosphate, and it becomes glucose, the reverse pathway, and goes to the circulation to give energy, because energy to the 5 main things, that is CNS, central nervous system, RBC, testis, kidney, and what else?
Retina.
Retina. Yes. So this is how lipolysis happens.